afternoon. My name is Marta Dominguez, and today I'm going to share with you my thesis research, which was about incorporation and effects of microplastics on seahorses. Let me start by giving you some background information about microplastics and the species of the study. So microplastics represent a significant hazard to marine ecosystems, and although the global concern has increased over the last years, the effects they have on organisms remain, and the long-term consequences remain largely unknown. These synthetic particles are smaller than 5 millimeters in diameter, and they can be formed from a wide range of components. According to their origin, there are two types of microplastics. Primary microplastics have been made with a small size to be used in different applications, and secondary microplastics result from the hydrolysis and biodegradation of larger items under environmental conditions. Aquatic organisms can either directly ingest plastics due to the confusion with their food, or through indiscriminate feeding behavior, or indirectly by trophic transfer when they eat a contaminated prey. Their small size allows them to enter and be transferred along the trophic chain, with the potential for bioaccumulation and the release of contaminants associated to these microplastics. Acartia is an ecologically relevant copepod species, and it's widely distributed and located at the base of the food web. Therefore, the accumulation of pollutants by this species poses risk to its predators. Hippocampus reidi is a species of seahorse with a small home range and resides in warm, shallow, tropical waters of coastal habitats. This species is listed in the IUCN Red List of Threatened Species. They are very sedentary fishes since their swimming capacity is very poor, and this attribute increases the risk of exposure to pollutants. Some of the threats of seahorses are the degradation of their habitat, the illegal trade, and the gathering of millions of individuals per year to contribute to traditional Chinese medicine. Seahorses can be used as bioindicators of contamination. Therefore, this group is of great ecological importance, and although little is known about their population ecology, distribution, or behavior, they have a great potential to act as sentinel species. Nowadays, the impact of microplastics on seahorses remains unknown. Therefore, the aim of this research was to study the ingestion and assimilation of microplastics by the copepod Acartia and by the seahorse Hippocampus reidi. Specific objectives were to evaluate the ingestion and residence time of microplastics in the prey of the seahorses, Acartia, and to evaluate the ingestion and residence time of microplastics by seahorses using Acartia as a transfer vector. Let's go to move to materials and methods. First of all, it is worth mentioning that fish handling and sampling were conducted in compliance with all bioethics standards on animal experimentation of the Spanish government. The present study was conducted in the seahorse nursery located in the laboratory complex of the Marine Research Institute in the north of Spain. After hatching, a total of 600 juveniles of Hippocampus reidi were obtained, and they were maintained in treated seawater at 26 degrees and 32 salinity. Seahorses were fed twice a day with leaf copepods of the species Acartia, and these copepods were cultivated at 26 degrees and 38 salinity, and they were fed on mixtures of the microalgae Isochrysis galvana and Rhodomonas lens. The microplastic used for the experiments consists of polyethylene microspheres of 1 to 5 micrometers in diameter, stained with red fluorophore. For the experiment 1, which is related to the first objective, copepods were cultured in a 500-liter tank, from which 8 liters were taken and placed in 4 bakers of 2 liters each. Copepods from 2 of the bakers were fed with the algae mix, and those from the other 2 bakers were fasted for 6 hours. Copepods were exposed to microplastics for 1 hour at 2 different concentrations, 10 and 100 micrograms per liter. After the exposure, 1 liter from each treatment was filtered, and copepods were resuspended in bakers with clean seawater in order to remove all microplastics that had not been ingested. Samples of copepods were sequentially taken during 2 hours at different times, and later they were fixed for further observation under the fluorescence microscope. Regarding the experiment 2, the remaining litter of copepods from each treatment mentioned in the previous experiment was also filtered, and it was used to feed the seahorses, juvenile seahorses. And the seahorses were fed with 1 liter of fasted and fed copepods. Subsequently, 3 seahorses were collected from each baker at 20 and 60 minutes of exposure. They were fixed in order to be observed under the fluorescence magnifying glass. The skin of seahorses is not translucent enough to allow the visualization of microplastics under the fluorescence magnifying glass. Therefore, the pigmentation process was performed so that the coloration of the tissue was lighter enough to observe the ingested microplastics. All the copepods and seahorses were observed under the fluorescence microscope and the fluorescence magnifying glass, respectively, and some photos were taken in order to show visual comparisons. Also, microplastic incorporation rates of copepods were measured by comparing the photographs before and after 15 minutes of exposure. And the same comparison was applied to seahorses that were exposed to copepods for 20 and 60 minutes. Also, a statistical analysis could not be performed due to the lack of time to process the samples of seahorses and copepods, since the COVID-19 pandemic did not allow to finish all the samples. Therefore, the analysis was performed qualitatively using only the photos taken. Let's go to move to the results. So the retention time of microplastics in copepods. After observing the copepods under the fluorescence microscope, microplastics were observed at 0 minutes of exposure, but after 15 minutes, they show very few microplastics at the concentration of 100 micrograms liter and none at the concentration of 10. The photos at the top show ingested microplastics located close to the mouth, while the photos at the bottom, the microplastics are located both in the digestive tract and the anal segment. Interestingly, the presence of microplastics was higher in copepods that were previously fasted than in those that were previously fed with the algae mix. So the microplastics were only visible at 0 minutes of exposure and at the concentration of 100 micrograms liter in both fasted and fed copepods. Therefore, it was suggested that microplastics were evacuated after 15 minutes, since no microplastics were observed in the copepods that were kept under the same food and microplastic conditions. And we can see that in these pictures. Retention time of microplastics in seahorses. So after 20 minutes, seahorses that were fed with fed copepods showed microplastics at the concentration of 100 micrograms liter, which are the photos C and D. And there was only 1 seahorse that did not show any microplastics after 20 minutes of exposure, which is the one at the top, photos A and B. Among seahorses that were fed with fasted copepods, those exposed to the lower concentration of microplastics, A and B, presented a lower amount of microplastics than those that were exposed to the higher concentration, C and D, photos C and D. Seahorses that were fed with fed copepods also showed microplastics, which means that copepods feeding on the algae also ingested microplastics. After 16 minutes, fluorescence microplastics were observed in seahorses at both concentrations, regardless of copepods offered to them having been previously fed with the algae mix or not, or fasted. The picture on the left shows seahorses fed with fed copepods, and the picture on the right shows seahorses fed with fasted copepods. But in both pictures, microplastics are present. Generally, all the seahorses fed with copepods previously exposed to a concentration of 100 micrograms liter presented a higher amount of microplastic than those that were exposed to 10 micrograms liter. Moving to the discussion. So the present research shows that copepods have a high evacuation rate for microplastics, suggesting that their retention time is very short, since after 15 minutes, almost no microplastics were detected. Also, results reported here indicate that microplastics accumulate in the mouth, digestive tract, and anal segment of the copepods. Fasted copepods showed a higher ingestion of microplastic than fed copepods. This might be due to the fact that copepods are able to detect and distinguish between the algae and microplastics, showing a predilection for the algae. Regarding seahorses, this study also confirms the trophic transfer of microplastics, since seahorses ingested microplastics through previously exposed copepods that can be considered as the natural prey. There was only one seahorse that, from those observed, that did not show any microplastics 20 minutes after being fed with copepods, regardless the concentration. Also, seahorses have demonstrated to incorporate and accumulate microplastics, both in their digestive tract and close to the anus. It's been found that those juveniles exposed for 20 minutes preferentially accumulate microplastics in the digestive tract, while those that were exposed for 60 minutes accumulate them close to the anus, probably as a result of the digestion process. In the present study, a higher concentration of microplastics has been shown in seahorses that were sampled after a long period. And this might suggest that seahorses do not efficiently excrete polyethylene microspheres, since the digestive system is not fully developed, although further studies, of course, are needed. Seahorses then showed a much higher retention of microplastic than copepods that were able to efficiently excrete them. To conclude, this study provides the first estimates of the retention time of microplastics in seahorses, and it also reveals that there is a directly proportional relation between the time of exposure and the amount of microplastics in the digestive tract of Hippocampus reidi. The demonstration of the accumulation of microplastics in this species of fish is of great concern, as well as the transfer of microplastic with copepods as vectors. However, more research should be performed on the impact of these pollutants to seahorses. Also, further work will investigate the biochemical effects on copepods and seahorses of virgin polyethylene microplastics and the same polyethylene microplastics loaded with organic pollutants. The exposures were already carried out, but biochemical analysis could not be completed due to the COVID-19 pandemic. And this is why could not be included in this thesis, but now this work will be resumed. This research provides a study with a threatened seahorse species and provides useful experimental evidence to assist conservation and management initiatives. Since Hippocampus reidi is considered a good bioindicator of contamination, the cultivation, knowledge, and protection of this species is of great importance. Seahorses could thus become a good tool for monitoring microplastics in marine habitats. Thank you for your attention. I would like to thank my supervisors Jorge Hernandez and Juan Bellas, my promoter Miquel Planas, and to everyone who was involved in my project. Thank you.